All right, good evening, everybody, and welcome to our Topics in Regenerative Medicine lecture series. The lecture series is supported by a grant that we have in conjunction with UC Davis from the California Institute for Regenerative Medicine, or CIRM, and also by the Sac State Center for Science, Technology, Engineering, and Mathematics, or STEM Excellence. So I'd also like to thank our partners from UC Davis who have really helped us in putting together such an exciting program. I see Jan Nolte here and some others, and we're just very excited about this program and its continuance, and you guys sitting in these seats makes it look pretty likely to continue. All right. So I'm Professor Tom Landerholm from the Biology Department here at Sac State, and it's my pleasure tonight to introduce our speaker, Dr. John Chapman. John is a kind of a unique and fascinating combination of cell scientist, device engineer, entrepreneur, and his main objectives that he'll speak to you about tonight are to try to put together devices wherein scientists and doctors can take your own stem cells, your healthy cells, concentrate them, isolate them from the other cells, do whatever genetic analysis and manipulation that needs to be done, and put them back in you to address your specific biological and health needs. So it's personalized stem cell medicine. I just got to say, wow, there are just so many cool ideas in there. I don't even know where to start. So we'll let John take care of that. So in a minute, after I tell you where John's been. All right. So where has John been? John got his BS in chemistry in 1976 from West Texas A&M University, a PhD in toxicology in 1982 from the University of Arkansas for Medical Sciences in Little Rock. After a couple of years postdocing, John spent 16 years with Baxter Healthcare, the last 4 as scientific director. He then spent 6 more years as Vice President of Research and Development of Vitex Corporation and 5 years as the Vice President of Scientific Affairs and Business Development at Thermogenesis here in the Sacramento region. In 2010, John founded and now serves as President and CEO of Stem Cell Partners, also here in Sacramento. In addition, John is an adjunct professor of electrical engineering here at CSUS, an author on more than 40 peer-reviewed publications, and is named as an inventor on more than 20 patent applications. So without further ado, please help me in welcoming Dr. John Chapman.
All right. Well, hello, everybody. It's good to be here. Tom, thank you for the very gracious introduction. I'm talking today about an area that's really an exciting field of development. It's exciting because of what good it's doing today, and it's especially exciting what good it's going to bring tomorrow. And I see so many young people here, and it's a good area for you to think about pursuing, because it's a chance in the next 20 years to see how healthcare will change from being just not only a pharmaceutical-based approach, but using cells as a therapeutic agent. And that's what I'll be talking to you today about. Let's see. Okay. Did I change? It's my own computer, so I can't say anything. Okay. Before I talk about regenerative medicine, I want to talk about generative biology. To think that all of us begin our lives as a single cell, and after a few multiplications, you have an embryo, but subsequently, that one cell will become 100 trillion cells. That's a big number. That's bigger than our national debt. And that's a lot of replication, and it's high fidelity replication. It creates 206 bones, 600 muscles, and 22 internal organs. That's a lot of differentiation, a lot of functionality. The average human brain has 100 billion nerve cells. I stress 100 billion is for average, so you know, you young guys and gals have fun, so you may be occasionally dipping in below 100 billion, if too much. But I'm sure we're going to regenerate back. So now I want to talk about regenerative biology. And I think the first point I want to make is, before we talk about regenerative medicine, we need to understand that our body is not static. We're changing all the time. For example, that there's 300 million cells are replaced in our body every minute. So we're constantly in a very dynamic state. The skin that you look at, that skin was not there 2 weeks ago. That's new skin. Well, and we change our skin about 27 days, every 27 days. So over our lifetime, we'll have 1,000 different skins. So we're constantly regenerating. That's just normal biology. Our stomach mucosa regenerates every 2 weeks. I guess, living in an acid bath, I probably would want to regenerate every 2 weeks too. Our taste buds regenerate every 10 days. And the reason for pointing this out is to say our body regenerates itself naturally. It's part of our normal being. But nobody is king of regeneration than the bone marrow. The bone marrow is kicking out 2.5 million red blood cells every second. Well, there's another 2.5 million. What did you do? So that's an incredible productivity. Those cells go into circulation. They last 120 days. Then there's other cells called neutrophils, which are important for fighting bacterial infections. The bone marrow produces 7 million of those cells every minute, and they only live for 2 to 3 days. And in our body, we have about 1.5 gallons of blood. And over our lifetime, we'll generate more than 300 gallons. So that's a lot of blood. Now let's talk about regenerative medicine. And this is where it really gets cool. Because this slide is showing some homegrown stem cell technology. This is the technology that came from thermogenesis. The inventor is Phil Coelho, lives here in Sacramento. He's a good friend, neighbor, and very creative engineer. And so in this process, you have the baby being born here. The placenta is put into a basket. The umbilical cord is hanging down. A phlebotomy procedure is done on that vein. And then it goes to a device that is manufactured by thermogenesis called the AXP. It will fractionate that blood, cord blood, into a plasma fraction, a packed red cell fraction, and a stem cell fraction. So here is the stem cell compartment containing the stem cell cord blood concentrate. This is where the stem cells are. It's put into a cryo preservative device. This freezes the devices in a controlled way. And when it stays in liquid nitrogen, which has a temperature of 196 degrees, minus 196, and it'll stay there in a suspended state. So there is a way to stop time in terms of biological time, and that's to put the cells in such a cold temperature. And so here we look at a frozen unit. It's ready to be frozen, I should say. This bag here contains 20 milliliters. It's like half a cup. But the power of it has is really, truly awesome. Here's a young boy. His name's Ryan. In 1988, I believe it was 88 or 89? 88, yes. He was the first person to receive a cord blood transplant. And before that, bone marrow was used, but now they're using cord blood. So that half a cup of blood that was in that bag, that amount of cells was transfused to Ryan because he had a serious disease that would have been fatal for him. And now he's this age, doing well. So all the blood in Ryan's body was derived from that 20 mil aliquot. Those stem cells that were in that bag, which could be easily contained in the size of a BB, are what's keeping Ryan alive, and it's replicating, and it will keep providing him all the blood that he needs for the rest of his life. So now, I was talking about the generative power of biology at the beginning, talking about how we go from 1 cell to 100 trillion. And then I talked about how we normally regenerate our skin and mucosa and the blood, but now we're harnessing that therapeutic potential, that regenerative potential to provide a medical care for patients. And so this is a very good example of the power. And what we're trying to do as scientists, engineers, and physicians is harness that power. We're trying to learn how to do that. And that's the effort that we're doing. If someone is interested to learn more about cord blood, there's a book called by Stephen Sprague, Beyond Leukemia, The Conclusion to a 10-Year Story of One Man's Cord Blood Miracle. Here, this is Stephen here. This is another woman who received a cord blood transplant successfully. And this is Dr. Pablo Rubenstein of the New York Blood Center. And he has really been pioneering the field. And he's been a great friend of thermogenesis throughout the years. So let's talk about regenerative medicine. The regenerative medicine can be defined as using the living cell as a therapeutic agent It offers a radically new and powerful means for delivering improved, cost-effective health care. And I'm going to talk, that's what really the talk is about. I think no one is satisfied with the state of the health care. People, it's too expensive, we can't see the doctor, it's difficult, but a lot of times they just don't have the care that we need. There's so many conditions that cannot be treated. And sometimes we feel like the treatments are only temporary and relieve the pain, but they really don't resolve the problem. They don't really get to the point of being a cure. And many times the patients don't respond to the treatment that we want. A great drug, if 30% of the patients respond to a pharmaceutical drug, that's great. It's a great drug, 30% response rate. So there's also problems with pharmaceuticals having significant side effects. And of course, the high cost per treatment is really threatening the healthcare system, especially as us baby boomers come in to take the money in the Medicare system. So I want to talk about regenerative medicine versus pharmaceuticals. In regenerative medicine, which is the use of cells as a therapeutic agent, what we do are trying to promote healing by the living cells. There's fewer treatments and they have prolonged effects, but they need to be personalized for each person. Whereas pharmaceuticals use molecules, so it's a molecular approach versus a cellular approach. They provide transient effects, and they're pretty good at reducing chronic risk, like reducing the levels of cholesterol in your body. So they do have good effects, but they tend to also have side effects that are of concern. And so the way I would compare pharmaceuticals and stem cells is the difference between a note and a music. What I mean by that is if you have a keyboard and a piano, a drug is like one key on that keyboard. If you want to increase the dose, you hit it harder. You want to give it fast, give it more often, you can get a bigger effect. So, but that's only hitting one note. Whereas stem cells are really like playing music. They can change, adapt to the situation. So they know the difference between playing Christmas music and playing birthday music. They understand the environment they're in and they respond accordingly. So we and especially you, since you're younger, need to learn how to harness this incredible therapeutic potential that stem cells have. And I think that's going to make all the difference. So I want to talk about two types of regenerative medicine, the here and now and the then and wow is what I say. So that here and now is promoting angiogenesis with autologous stem cells. Autologous stem cells is a word that just means using your own cells. But there are many promising things that are going on in stem cell therapy that are just incredible, and not just only using your own cells, but growing cells in the laboratory, taking your own cells of your body and inducing them to become pluripotent stem cells. So there's a whole world open to you as young scientists that you can enter into and drive this whole regenerative medicine area. So examples of the then and wow are the research that's going on now. If you look at a website called clinicaltrials.gov, that's a website that is worldwide reporting of all the clinical trials going on. So you can go in there and Google stem cells in heart or stem cells in pancreas, stem cells in anything, and it'll give you a listing of all the clinical trials that are going on in the world for that area. And really every major organ system is being investigated. It's a tremendous amount of research activity going on, and a lot of it's going on right here in Sacramento, homegrown stem cell technology. I'm very pleased that Jan and some of her team Jan Nolta joined us. She's at the Institute for Regenerative Cures, and that is a wonderful place for research, 150 physicians and scientists working together to do these kinds of advanced stem cell treatments. And it's very exciting. So I'm 57 years old, okay? So I got a biological clock ticking on me. And so I'm trying to focus on things that are going to happen in my time that I have left to be a productive citizen on the planet. And so I'm focused on autologous cell therapy, meaning this is the process where you take the cells out of a body, you isolate them, you can activate them, and then you deliver them back to the patient, all within the same surgical procedure. So this is a translation issue. where we're learning we're seeing what the basic research is doing, and we're figuring out how can we make it easy, quick, fast, not expensive for the doctors to do this in the operating room setting. This is commonly done by orthopedic surgeons, and so it's not the biggest market out there, but it's $100 million in market that's being sold in orthopedic. And what the orthopedic surgeons generally do will take stem cells from your bone marrow, combine it with a bone graft material, and the combination of the cells and the bone graft can be placed where they want bone to grow, or they can cause the bone to heal faster. And so that's really a commonly used method for using stem cells today. So what I think is really an exciting understanding is that another thing that those same cells that grow bone can do is that they can promote growing new blood vessels in the process called angiogenesis, or neovascularization. And it's a very complex process, and the way that it works is dependent upon the interaction of a lot of cells coordinating with each other. They're secreting cytokines, they're secreting trophic factors, but collectively what it causes to happen is a wound or an injury site to become vascularized faster. And that is a tool that we can use today to help patients get better. And I'm going to talk to you about how that's being done. First, I want to point out that bone marrow, injecting bone marrow cells back into the patient has an excellent safety track record. There's no incident of anybody having any side effects or being harmed by this. So, but it's very important that these trials be controlled, because we have an excellent track record, but you know, you cannot put any cell to anybody at any time, in any place, if any pathology. It can cause harm. There will be, so there has to be good judgment used. And that's one thing that we really, as scientists, clinicians, and engineers, we all depend upon each other to be responsible and work in a good way that will keep the field moving forward. So I'm going to talk now about a disease that I care a lot about. It's called critical limb ischemia. It's a disease that has affected my family, and it's something that will probably affect your parents first. It's a disease of older people, but it's something that it's important to understand. Atherosclerosis is the buildup of plaque material into the blood supply. So you can see the blood that flows normally without that, but as this plaque accumulates, it will eventually block that vessel, and now you have restricted blood flow. And so as a result of that, you set into a sequence of pathologies, which can first start as being limb pain. The patient, it hurts when he walks, and then as the lack of blood flow becomes more severe, you can have functional loss where they can't really walk any distance at all. Gangrene can come, and then you can have amputation. So this area of diseases is called ischemic diseases, and the one that we're talking about today is peripheral artery disease, but it's related to cerebral vascular disease and coronary heart disease. All of these are in the same family of treatments. It affects 8 to 12 million people, and 1 in every 5 people over 70 will be affected with this. And particularly at risk are patients who have diabetes. And so that's important to control. Other risk factors for developing peripheral artery disease are obesity, hypertension, aging, and smoking. All these things are things that can be controlled and should be controlled to minimize that risk. So what happens with patients who have peripheral artery disease? Most of them simply have a leg pain on exercise. So when they're walking, they feel like it's cramping, it hurts, and so they have to stop. Their mobility becomes limited. Some have what's called claudication. Claudication is where you have pain even when you are not exercising. You're just standing in the room or sitting there, it hurts. And then 5 to 10% will go on to have critical limb ischemia. So, and all of this is caused because you don't have sufficient blood flow down to the lower limb. Janice taught me the next, I've shown this slide before, and I was criticized for, you didn't need to show that slide, John. But I show it, and I'm showing it to you today, because it's a serious problem. And I want to show that we're talking about real change, real therapy, and helping people. So what can it be? So if you're easily offended or you don't like gruesome pictures, then I'll let you know when it's okay to look. but these are pictures. So what can start out as an ischemic ulcer for a person who has this bad flow of blood to the lower limb, stubbing your toe, having an accident when you're trimming your toenail, that little injury can start a cascade that is just horrific. It seems like nothing to anybody else. You stub your toe, but for that person, it can be the beginning of a very long and unpleasant trip. So you have sores, lack of blood flow, you have inadequate healing, the inadequate resolution of infection. So what happens? Gangrene sets in. And then because of a gangrene, that gangrene means that you have uncontrolled infection, bacteria infection, very unpleasant for the person, very difficult to take care of. The next step is amputation of the toes. And when that part of the body develops gangrene, they go to the ankle. And then it goes up to the leg. So it's something that I've witnessed, I've experienced, and went through with my mom. And so I know it's real and it's really bad. So it's fortunate that, we're working on ways to how do you intervene and you stop this. So what is the current treatment is there's ways to deal with this, both through exercise, medications, and there's revascularization procedures, there's some pharmacology. And what we're talking about now is in the surgery, they do bypass surgery, or you can do endovascular treatment by interventional vascular procedure. And then when that fails, it goes to amputation. So basically, what you're doing is you're fighting a fight that you're not going to win for a lot of patients, is that you can delay, but there's a process in motion that it's very hard to reverse. And so what people are working on now are the use of cell therapy intervention right at this point. These patients are called no-option patients. That means that they've exhausted all the other medical treatments available to them. The next step is amputation. I talked to a doctor, and Dr. Eaton in Miami he's one of these guys that when he's 16 years old, he's at MIT, and he's going to be a physics major. He took the first physics test, and he got a 35 on it, and felt pretty good about it because he saw the other guys had 25 for their score. But then he noticed there's 4 other people in class that got 100. And he said, wow, they got a gear I don't have. And so he became, he switched majors from physics to medicine. And I'm glad that he did. I met him. He's a great guy. And he was telling me about, I said, boy, doctor, the patients must really be disappointed when you tell them that you're going to have to do the amputation. And he says, John, you don't understand. They're in my office begging me for that amputation because it's how painful their life is. There's just no blood flow to that leg, and it hurts. So this is really a last step intervention that we're talking about before that amputation occurs. What are the goals of that treatment? Well, the first goal is to stop the pain. You want to make it possible for a person to start walking again. You want to heal the ulcers. You certainly want to prevent the amputation, and there's also complications of cardiovascular mortality. And you just want to improve the overall person's quality of life. Those are the goals of the treatment. And so these are the current treatments, the stent, balloon angioplasty, and bypass surgery. And one thing I want to make a comment that's really exciting about regenerative medicine, and one of the reasons why I'm very passionate about it, is regenerative medicine levels the playing field across economic barriers. There are places where people can afford to buy a $3,000 stent, but they can't afford to withdraw bone marrow aspirate and put it into the limb. So there's ability to do this in developing countries that's very exciting. So I have a friend, Dr. Brazzini, in Lima, Peru. He told me about the woman who had a sole source of money. She sold ice cream off of a bicycle stand. They have a bicycle with ice cream on it. She couldn't do that anymore because of the pain. And with his money, he did the treatment for her that we're going to be talking about, and now she's back and being able to make a living for her and her family. So this is something that can happen even in countries with restricted economies. And I think that's a very exciting thing about regenerative medicine. So the reason why cell therapy is necessary in addition to the endovascular procedures is what we're talking about. The endovascular procedures can go after the macro circulation, the big blood vessels. But once the capillaries start shutting down, then you're out of business. So the cell therapy can attack the micro circulation problems, and the physician can address the macro circulation. And really, they're complementary technologies. They have to be really done well together. And so there was a paper published in 2002. It's from Japan, where the investigators demonstrated the ability of bone marrow cells to restore blood flow to affected limb. So this is a laser Doppler, and so you can see the more blood flow is, there's color. And in this patient, the left foot, this is the unaffected limb, and this is the affected limb. So it's interesting, when people have a critical limb ischemia, it's not unusual just for one limb to be affected. It can be both, but in many cases it's one. And so you can see there's a difference in here, and that is due to the lack of color here is due to the lack of blood flow. So they injected the mononuclear cells into the patient's leg. 7 days later, they can see it coming back. 14 days later, stronger, and 28 days, very well. So this was a paper published in 2002, and it's a landmark paper. It's a very, very important paper, and it opened up a whole new field. And it was really exciting because there was some disappointment coming in the area because gene therapy had been tried using VEGF as a way to induce blood flow, and it wasn't working well. And then coming in, instead of with a gene therapy approach, coming with a cellular therapy, it worked much better. So when I visited Dr. Eaton, I went with him and saw the procedure. And so the way that he was doing the procedure at the time was taking 500 mils of bone marrow from the iliac crest, and then those cells were shipped to a laboratory in the hospital, and they're processed in this instrument here. The complicated instrument. You see there's a lot of tubing going every which way. It's a Cobe spectra system. It's usually used for separating blood for platelet pheresis. They have the product here, 2 billion cells in. And then what they do next is they take these cells and they make a grid on the patient's leg. This leg's ready for amputation. This is it. This doesn't work, it's going to go. So they make a grid and they put the marks. And then they do about 0.7 mil injection. So they take the needle, go almost to the bone, inject a little bit, pull up, inject a little bit, pull up, inject a little bit, pull up. And they keep doing that for all the whole range. And because the procedure takes 6 hours to get the cells back from the laboratory of the patients, is not really in the, they give them sedation for the bone marrow draw, but not because they're taking 500 mils of bone marrow. It's a lot. So that has to be done under anesthesia. But because it takes so long, the person's no longer under anesthesia at that point. And he now, can you imagine, he's injecting these cells into the limb that hurts so bad, you wanted to amputate it. And he said, well, they only feel about the first 10. And so, that's really pretty brutal, I think, to think that. And also they make some injections on the foot. So what happened? So 5 days later, the foot became warmer because the foot's cold. There's no blood flow, so it's warm, and they have less rest pain. 9 days, the right foot and left foot had the same temperature, but now they didn't have to take pain medication at night. After 23 days, they're starting to walk. The foot still has no feeling in it. At 52 days, they had no rest pain. They're walking 4 blocks. The sore on their foot healed. And now when they press on the foot, it doesn't have dependent rubor, meaning that there's enough blood flow, it can go. 70 days, that's what the leg looks like. Even, he says, interestingly, they can even see hair starting to grow back on the foot. I don't know what that means, but I guess it's good. But clearly, this was a limb that was destined for a trash can. And instead, it's on that person's body where they can be productive. And that's the power of regenerative medicine. And also, what I really like about critical limb ischemia as a starting point is that you can have objective measures of, is it really working? So we talked about the laser Doppler data. You can also do transcutaneous pressure oxygen meter reading. So you have totally objective data, and you can see that initially there's very low oxygen tension in the tissue, and with time, they can see it coming back. So again, this is not subjective data that is saying it's getting better. It's physiological data that says it's getting better. So here's some homegrown technology, Sacramento. This is a photo of my colleague, Dr. Vijay Kumar. He co-invented with me a product called the Rescue System. And this is the system here. And what that system it does is it enables the bone marrow, I should say, to stay in the operating room. And it processes it within 15 minutes. So now those cells don't have to go to the laboratory. They can stay in the room, and they can be concentrated there, and they can be delivered to the patient while he's still under anesthesia. So when he wakes up, it's over. So this is the technology that we're doing right here in Sacramento. And, you know, and part of it is just there's important clinical trials also going on. I think you're starting a new study, right, Jan? There's a new critical limb ischemia study starting. So this is a very active area, and we're excited to be a part of that. And I'm excited to have been a part of that project. So the goals of the treatment with the critical limb ischemia were met, all of them. Now so far I've told you one study, and I told you one patient anecdotal study. But what's happened is that once people learned about this, these results have been repeated in 19 clinical trials all over the world. And you can see what the endpoints are. We're looking at how does the patient respond. And then they categorize it as either was it successful or neutral or did it fail. So red would be fail. But these studies were found to be equivocal finding. Wasn't sure if it worked on only 2, but in 17 of the 19 studies, they were able to confirm the findings of the Japanese investigators. So that's very exciting. Also, it can be performed not only using bone marrow cells, but also mobilized cells from the peripheral blood. So that's another way it's been shown. And then it's also been shown in animals to work. So Jan and her team have an animal model that she can recreate this disease state in laboratory animals and show that they can respond. So here we have preclinical animal data and reproducible results in the clinic. So this is real. This is a real effect, and it's working by causing blood vessels to grow. There's still a lot of work to be done before it becomes routine therapy. These all been done on no option patients. We need to know what's the right cells to give, what fraction. Should we purify them tremendously, or is a Buffy-Co good enough? So there's a lot to learn about that. Can we use FICO when we prepare them? So there's some methodology to work out in terms of understanding what is the best way to prepare those cells, what's the best route. You can give it intra-arterial or you can give it intramuscular. How many cells do you need to give? The cell dose response is not well understood at all. So we need to understand that. And there's also the possibility of helping those cells work better before we give them to the patient. And this is an area of research that I'm very interested in, and it's called preconditioning. So this is doing where you take the cells out of the body, but before you return it to the ischemic tissue, to the body, to do a preconditioning step where you prepare those cells to maximize their therapeutic potential. And so I'm working with Jan and her team. We're figuring out the way to get studies, honestly, the way to get studies funded. We know what to do. We have you have to have the money to do that. But we're excited about the concept of what we're going to do to make the therapy even stronger. And so we're looking forward to doing those experiments. Okay. So now let's talk about what's involved in autologous cell therapy, because the purpose of really showing this is to talk about what kind of jobs could you have if you wanted to be a part of the regenerative medicine space. So the elements of regenerative medicine are devices for cell processing. You have bioactive agents such as cytokines. You have cells. You have biomaterials that can be scaffolding. So really, there's so many opportunities to learn how do you make the cells work better. How do you present them to the body is very critical. So the scaffold, how do you put the cytokines to stimulate the maximum effect? So really, we're talking about building biocomposites consisting of cells, materials, and cytokines, and bundling all that together to create the desired effect. And that's work to be done, and it's all promising opportunities for employment, in my opinion. Another part of my presentation was, they said, well, John, don't just talk about dead legs, okay? That's kind of morbid a little bit. Another place where they're doing moving cells, taking cells out of the body and putting them back in is in the cosmetic surgery area. So here, if you do a procedure called liposuction, you have a cannula go in, and it'll aspirate the subcutaneous fat, and when you pull it out from different regions of the body, you pull here, and you end up with a canister. It looks like a milkshake, strawberry milkshake. Hey, it is what it is. and believe me, when you try to pipette that, it is like a strawberry milkshake. I mean, that pipette gets hung up pretty badly. So what's needed and what people are working on are ways to process this adipose to make it become a fat graft. So now you have a biological cell mixture, and you can put that in the body wherever you want to increase volume, okay? And so I'll leave it to your imagination. Where do you want to increase volume? Personal choice. So, but anyway, when you concentrate it, this is what it ends up looking like this. So it's like a material that is a solid. And the trick is, how do you retain the volume? How do you retain the volume of cells that you inject? Because right now, the challenge is maybe you can retain about 50% of the volume that goes in. Well, what's needed is a way to improve the survival of those adipocytes that are going in. And so, and I should say, it's not only just for cosmetic purposes, but there's also some reconstructive surgery that's done that's very important to helping people have helping people have good lives who have been really debilitated by a lot of things. People in the military are working to help soldiers who have been injured with blast injuries, and it's just tremendously impressive to see and hear from the soldiers who are going through their injuries, and to know that there's ways that regenerative medicine can reach out and help them get back to have the best quality of life possible that they can have. So it is for plastic surgery, but plastic surgeons don't just do cosmetic work. They also do reconstructive work, and I think that's important. So in my view, right now we have what I call an adipose technology gap. That means on one end we have the super duper machine that's very nice. I mean, it does it all, but it's very expensive. It's a $100,000 instrument. Each disposable costs about $2,000. So that's on the high end. Then on the other end, we have the doctors kind of doing it themselves. So, you know, it's that kind of openness is that's shocking to me. Coming from the blood world, we like to keep everything closed. We don't like the atmosphere to contact anything. And so seeing an open system like that, I felt like the area had been neglected. So there's a sweet spot to be found to have a technology. And so there are people working. It's a little bit of a gold rush going on there now. People trying to develop the right machine to do that and right technology, because we think adipose cells are actually probably going to be the most important source of autologous stem cells, because, well, we all have more than we need, right? And it's a lot easier to give than a bone marrow tap. So now I'm going to talk about careers in medical product development. I think one thing that's unusual about my being here is that I'm an industrial scientist. I'm not an academic scientist. I came straight to the world of industry right after getting my PhD. And so by understanding the process in which medical products are developed, I think you'll see the opportunities that you can have if you want to have a career in the medical device industry. And I would encourage you to consider that. First begins with the selection of a problem. The problem that really takes the prepared mind. You have to be reading the basic research. And what we try to do is really translate what is happening in the basic research in the universities to understand what's happening in basic research. How can we apply that to the clinical setting? And so when scientists think they have an idea, they'll talk to a person in the marketing function. And the marketing person will tell you if there's any financial incentive for the company to do that. So having a science background and going into marketing, it's a good career opportunity. We need that. We need people in marketing who understand the science. That makes a good partnership between the scientific team and the marketing team. So that's if you're thinking about doing science, think about a career in marketing, because that may be what you're very good at. Then once you know the problem, you have what I call the creative sparks. This is actually a plug for a company. Creative Sparks is the name of the company. And Rodney Sparks is the person, it's his company. Rodney Sparks is a graduate of Sac State. He and I are working together. He's a very, very capable, inventive mind. And I think Sac State should be proud to have him as an alumni, because he's really quite an impressive fellow. He works on fuel cells, he works on ophthalmology projects, he works on medical devices, and that ability to go across the spectrum of industries where you're cross-fertilizing, I think that's what makes him really effective. So it's good to be well-rounded, I would say. So once the customer requirements are defined, then the biologists go to work, and we define the process. So the biologist will come up with a protocol. So we're going to do adipose. Well, we want to make single cell stem cell suspension, so we need to digest the fat. We have a protocol. You add the collagenase, you centrifuge, you pellet the cells, you wash the cells, and then you have to recover those cells. Then we'll hand that over to engineers, and those engineers will be mechanical engineers, electrical engineers, firmware engineers. They'll make a machine that does that protocol. And then after they finish that, they'll bring it back to us, and we'll test it, and we'll see how it's working, and we work together to get that right. So I was jokingly, and I didn't think of it as an offensive thing, but I said one biologist will keep 5 engineers busy. And it's true. You know, normally in a company, I've always been an island of biologists surrounded by a sea of engineers. And it's just the way it is. I didn't mean anything negative to the engineers by saying that. Another thing, then you have proof of concept prototyping. So there are companies who are very capable of doing that. And actually, that's another industrial revolution that's occurring. If you can think of something, you can have it made in your hand for a very small amount of money, really, $300, $400, and it can work. So everyone can be an inventor now. Don't limit yourself to thinking, well, inventors are, I'm not able to do that. It's something that somebody else. Learning about inventing, learn about patenting. These are opportunities for yourself that you should at least understand and learn how prototyping works. So after you go through this process, we go for verification and validation studies. So we need scientists, again, to do all the studies to make sure the product's safe, make sure it's effective. A very good career opportunity is regulatory. Regulatory is where you represent the company to the regulatory agencies of the world. So you help the company get its products approved and registered. So that's a nice career to have. And another career you can have is quality assurance. Quality assurance is the group of people who are monitoring the company to make sure it's in compliance with what the FDA wants to do. That's a good career. So if you get a degree in science or engineering, doesn't mean you necessarily work in the lab. You say, I don't want to work in the lab all my life. Well, okay, but there's a lot of other kinds of careers that you can do in the medical device industry that you may enjoy and find enriching. And so just to sum up here on the last slide, I want to point out the relationship between the government, the universities, the industry, and investors. How do we all work together? And how important it is that we help each other and we collaborate and be a team. Because the grants that the academics get to do their basic research, what they're doing is converting dollars into knowledge. And that's that curve. The government gives them money, they return knowledge. How do they give that knowledge? They give it in the form of publications. Okay? So that's the transaction that occurs. What industrial scientists do is take that knowledge that's generated and do, by innovation, come up with products and patents that generate dollars. Well, Uncle Sam is waiting to get his share of that revenue, and he takes it. But he takes it and gives it back to the universities, and so it flows. And then there's also investors here who are helping companies to do this. So they invest money to create products, and then they get their money back. So it's just very important that we work together to make the cycle go. And as a country, I've traveled all over the world. I've been everywhere. And one thing that Americans can do better than anything else, anybody else, is innovate. We can compete based on our innovation. And this is a real economic test right now that we're in, and it's very competitive. I was interested in hypoxia research. I learned that in China, there is a whole building dedicated to hypoxic research. So the other countries recognize the importance of regenerative medicine, and they're competing. They know they can't compete in pharmaceuticals. That battle is gone. They've lost that. But they're engaged in winning the battle for regenerative medicine. So we have to be engaged. And I tell people, our competition is not in Sacramento. We're on the same page. We have got to work together, because our competition is not in Sacramento. Our competition is overseas. And so we have to work together, and I'm really happy how we're working together. I started a company here, and I couldn't be happier to be here because of all the support that I get. And I'm really appreciative of all those people who are helping me with that effort. Okay. I don't know what time it is. I feel like I've talked a long time. But I appreciate your coming tonight. I hope that it was helpful for you. And if you have any questions, I'll be here. And I wish everybody good luck on their finals. I understand it's final season.